Regarding my presentation, at first I didn't have a thing what to present at the today's workshop. So, so he gave me a vague idea about the tigers in Tarai. But Baburam has already told you about much of the conservation effort that Nepal is doing so far. So I'll be much more focused on my PhD research, which I did in the last 2 years. So I'll be presenting one of the paper from that. So keeping this slide, you can see that whatever the conservation challenge and the management goal in, as told by the previous speaker can be depicted in this slide. What we want is a protection of our habitat, where both all the animals roaming in our protected areas should roam freely, and our habitats should be protected. So that's all about the tigers in Tarai. So I'll be much more talking about our paper, which we did with the Nepal Tigers Genome Project. So basically focusing on the genetic structure and the gene flow of tigers in Tarai Arc. Just to give you a background, like in the recent times, the non-invasive method has been very much powerful in knowing how our population is doing. If you cite an example in Nepal, like the camera trapping survey, we have been doing a lot over the past years and without getting to capture the tigers. So we really know what's our population right now, how our population is doing in our protected areas. So in the recent time, using a genetic approach is also becoming a very much powerful technique. And it's gaining a lot of attention these days, especially worldwide. In Nepal, there are very few studies being done. If you cite about our flagship or the keystone species, like for example, there has been a study with the elephant in Bardia National Park, and also the snow leopard. There's a lot of work going on, and there's a paper published as well. And with the tigers, so we are doing the first genetic study on tigers. So whatever information we have got, I try to summarize these major findings and try to present over here. So what we know about the meta-population structure of tigers in Nepal, as Baburam already told, like across the Tarai Arc, we have our major tiger population is focused in the Tarai. So one study has been done based on the how the Tarai Arc was defined was based on the demographic population. So they have identified 3 clusters of a tiger across the Tarai Arc. One is Chitwan complex, one is Bardia complex, and the third one is Sukla joining all the way to the India. So we have basically a 3 meta-structure of tigers across the Tarai Arc in Nepal as well as in India. And if you talk about our corridors, so we have identified the major corridor across the Tarai Arc, and so over the period of a time we have been monitoring these corridors, and we know the tigers are well dispersing in between the protected areas, and we know that our corridors are very functional as well. So keeping this assumption in mind, so we have focused whether the tigers from Chitwan National Park is going all the way to Bardia National Park or not, whether the Bardia tiger is going to Suklavata or not. So we are using this genetic approach to measure some of these, our preconceived assumption. So our main objective was to define the genetic diversity and see the structures and the gene flow of tigers across the Tarai Arc. So one of the basic tool we have used is using the scat samples, because it's very easy to get and extract the DNA out of it. So the government of Nepal is doing a tiger census every 5 years, and also one of the main objective is to measure the occupancy of a tiger across the Tarai Arc as well. So we use the same methodology to collect our grid as well. So we focus on 30 grids from protected areas and the 10 grids from the corridor areas, and we try to find as much as scat sample as possible. So we did the opportunistic survey within each grade, and total we collected around 770 samples collected from across the four protected and the corridor area. Just I'll give you just a brief what we did. We first screened for the positive samples, then screened for the sex identification, and whichever the positive samples, we also screened for the, then we did for the individual identification. So whatever the loci we used in the individual identification, so we gathered the published results and tried to get, screen the loci out of it. So our basic knowledge we had is only from the outside perspective. We don't have any information from the thorax so far. So we try to gather the information what has been already been published, and we also collaborate with a few of the lab across the India as well, and try to screen only those loci so that our data could be comparable to them as well. So we screened 10 loci based on heterozygosity and the allelic diversity, and in the process, we calculated different metrics for PID sets for determining how much loci we need to individually identify the population. And we did the basic metrics before analyzing in detail about the like a Hardy-Weinberg equilibrium and the linkage distribution. So these are the few metrics we followed. Like for genetic structure, we used two methods. Population-based measures and the individual-based method using those software Harlequin structure and the tests. And we did the assignment test using a gene class and the structure, tried to see how our sampling get fit into those population or not. And because we are also interested whether the tigers are moving in between the protected area, so we also try to find the migrants in the population. So these are the few software which we use for the detection of migrant. And one of the recent migration rates we use using the software called the Bayes ass. I always like to tell this software because it's called the Bayes ass, right? So we also try to see if there's a bottleneck signature in our population or not. So I'll be presenting briefly. So these are the two methods we used. And also try to relate if there is a bottleneck signature, what might be the cause for it. So we did some lineage change analysis as well. So out of the 770 samples, so 51% was the tiger positive samples. Our amplification success was 85%. So two of the loci didn't work well, so we use the 8 loci information for the individual identification. Giving this graph, so we need a minimum of 6 loci. So we have enough power for our individual identification. So together we identified 78 tigers using 8 loci genotype information. And based on the positive samples, we have 40% genotype success. Because it was very, we did this survey during the winter and dry season, but we couldn't get a very much good sample from the corridor area. So we failed to genotype any samples from the corridor area, but the effort is going on. So 2 of the loci were out of the Hardy-Weinberg equilibrium, but it was not consistent among the population, so we could further do the analysis on our data set. So talking about the heterozygosity, so Chitwan was very rich in genetic variation, whereas very low in the Cyclovata population. If you talk about the whole of the thorax, so it was a moderate level of a genetic variation in our tiger population. So if you try to relate this with the globally, so in Nepal, like across the thorax, it's 0.61. If you talk about the tigers in the whole of the India, Italy, we are a little bit behind the Indian subcontinent. So if you talk about the tigers from the eastern, Southeast Asia, so it's around, it's a little bit, we are a little bit high. So if you talk about the global tiger population, so we are well below the genetic variation across the global population. Talking about the differentiation, if you talk about the Suklafata and the Bardia, there's a high level of a genetic differentiation. So if you have a high, so there is a less chances of a gene flow in between the population. So if you talk about the Bardia and the Sukla, it's relatively less level of a genetic variation. So if you talk about the Sukla and the Bardia, it's really moderate level. So one interesting to see is, although Chitwan and the Bardia are spatially apart, level of a genetic differentiation is really low. Whereas in between the Suklafata and the Bardia, although they are close enough, the level of differentiation is high. So one perspective what we can gain is Bardia and Chitwan were very much closely related historically. Right? So if you talk about the whole, like there's a moderate level of genetic variation. So giving the overall crux of this slide, so our conservation efforts should be continuing because we want to have a FST value which measures the level of a genetic differentiation should be as low as possible. If you talk about the genetic structure, so is there a three population of tigers across the Thapa arc? So we did a structure analysis, so using the program structure and the test. So these both result tells us that there is a three clusters of a tiger across the landscape. So that is Chitwan and the Suklafata. I'm not talking about the Thapa arc as a whole because we don't have a data from being inside of Thapa arc. So this is the plot diagram. You can give each bar plot gives you one individual tiger. So we have 78 tigers. So this also tells, this is the output from this true program. So this tells that we had three clusters of tigers across the Thapa arc. So this is the assignment test. What assignment test does is we collected the samples. So we want to know whether that samples belongs to which population. So altogether around from 78 tigers we have identified. So our assignment test result was 81%. So 81% we can correctly assign that particular tigers to that particular protected areas. And also the structure told us about the 73%. One of the management implication which we gain out of this slide is, if you want to do the genetic study in the future, so we can increase the number of loci to increase our correct assignment test as well. I'm giving you an insight, like you can see in the Bardia population, so 21% is the admix and the 72% is the resident samples which we got. So you can see the Bardia is a very good population from the genetic point of view because there's a lot more admixture going on from the surrounding population as well. So this is the detection of the migrant in a population. So we use the two methods, gene class and the structure. So from the indirect estimates, we found 2 migrants per generation. Per generation in the sense with the tigers is 5 years. So altogether we find around 10 migrants across the landscape. And one interesting result to see is between Bardia and the Chitwan, it's a male which is going on, whereas between Shukla and the Bardia, it's mostly the female tigers that are moving. So I like this figure a lot. So you have a Chitwan, you have Bardia, you have a Shukla. So this is the recent gene flow. So that means 5 or 6 generations ago, so around like 20 to 25 years ago. So it gives you a very recent data. You can see, as I already told, Bardia is in very good situation because if the tiger has to go to Suklafata, you can't go directly to Suklafata because the migration rate is too low. So if it has to go to Suklafata, it has first to go to Bardia, then to Suklafata. So overall scenario is that Bardia population is gaining more tigers from Suklafata as well as Chitwan. Whereas the Suklafata is giving more tigers than receiving it. So this is the kind of gene flow going on across the Thapa arc.
You have 5 minutes.
Okay. So and the other thing is about the second generation migrant as well. So we are getting a lot of tigers that are moving from Suklafata to Bardia. So we have also detected the bottleneck signature in our population. So that might be the reason we have like a low to moderate level of a variation in our population. So one probable cause may be the land use change analysis. Like Baburam already told you about how our protected area was established over the period of time. So before, like, tigers were allowed for hunting as well, and there was a lot of migration going on from the mid hills to the Tarai. So over the period of last 300 years, we lost 62% of our forest for agriculture. So those were the prime habitat for tigers as well. So that means 97% increase in agriculture in the last 200 years. So we also lost the tiger from the eastern part of the Nepal over the period of time. So now the tigers are confined within the protected area. So that might be the reason, the land use change, why there was a bottleneck signature across the tiger population in the landscape. So one closing remark which we found from this study was there's a low to moderate level of a genetic variation in our tiger population. So the genetic signature detected was due to the large-scale land use change. And yes, to our assumption from the demographic study as well as the genetic study, we have a genetic cluster of tiger in our population. And there is a high level of a gene flow going on between the in between the population, like for Bardia and the Sukla, and also from Sukla to Bardia as well. So these also relate to the importance of the corridor for allowing the dispersal as well. So just to give you an example, like India has recently declared the Pilibhit tiger range as a latest tiger reserve. So that also allows for us for allowing the gene flow of tigers from the Tarak, Nepal with the western Tarak population in India as well. So whatever we got from our result, the most important is whatever the conservation initiative we are doing in the landscape should allow for the gene flow of tigers across the Tarak. So with that, I'd like to thank Government of Nepal, CMD and Virginia Tech and the University. These are our collaborative effort. WWF and NTNC for allowing us the institutional support for doing some of the field work, and above all the USAID for funding the project. Thank you.